Good afternoon, everyone. Thank you for joining. Today, I'll be speaking to you about clinical pharmacology and how we fit into early drug development. Since the overall objective of my talk today is to explain a little bit about clinical pharmacology and discuss its critical role in early drug development, I'll be focusing on two main aspects. First, to understand the scientific underpinnings of clinical pharmacology as a discipline. And then, in the second part of my presentation, I'll be giving an in-depth overview of early clinical studies. There will be a focus on various aspects, such as study conduct, the information gathered as a result of these studies, and how to use that information in our drug labeling. And at the end, I'll also touch on a couple of developing areas in our field, including PBPK and model-informed drug development. Okay. So clinical pharmacology, what exactly is it? Like the old saying goes, if you ask 5 different clinical pharmacologists, you'll get 5 different answers. But in its simplest form, it is the study of the pharmacokinetics, or PK, and pharmacodynamics, or PD, of a drug in humans. PK is what the body does to the drug. It encompasses the processes of absorption, distribution, metabolism, and excretion for most drug products. PD, or pharmacodynamics, is what the drug does to the body. These two concepts are represented by these graphs on the right-hand side, where on the top graph you have a concentration of drug in blood or plasma, some type of biological matrix, over time. And in the bottom graph, you see an effect, some sort of biological measured effect over time. So what happens when we put it all together? We get a pretty powerful clinical pharmacology tool called a PK-PD relationship or exposure-response relationship, depending on the measured outcome. When we take a concentration over time profile and combine that with an effect over time profile, we get effect over concentration. So what we can really see is the effect of a drug over varying concentrations of the drug. And so in this way, we're able to predict some sort of quantified biological effect given any concentration of drug in the body. Given these tools at our disposal, how do clinical pharmacologists use them to contribute to the drug development process? Well, we like to say that we own the dose. And by that I mean we help determine the dosing regimen of a drug, how much to give, how often to give it. We even determine if a drug needs to be adjusted due to various intrinsic or extrinsic factors. In other words, are we giving the right drug at the right dose, at the right time, to the right patient? These are all very important questions that are asked of clinical pharmacologists and that we help to address through studies in early drug development. Now, I've mentioned ADME before, and assessing these processes gives us key information on the clinical pharmacology properties of a drug. The A in ADME stands for absorption, and it's simply the process of a drug reaching systemic circulation. It doesn't apply to all routes of administration. For an IV administered drug, there is no absorption since it's delivered straight into a vein and directly into systemic circulation. But absorption through an oral formulation, let's say, can be impacted by several factors in the body, such as eating food or pH-altering medications or other concomitant medications, alcohol, transporters, or drug metabolizing enzymes being altered in the gut. Important information that we typically communicate in the drug labeling, or USPI, in section 12.3 are the bioavailability of a drug, the time to reach maximum concentration, or Tmax, as well as the effect of food. The D in ADME stands for distribution. Distribution is the process of drug being distributed to different areas of the body, so it's just how it sounds. It is described by volume of distribution, which is a numerical descriptor that's given in the units of liters. It tells us the drug's propensity to either remain in blood or distribute to various organs throughout the body. So the higher the number, the more likely the drug is to distribute to various organs. Some factors that we need to consider for distribution are: does the drug bind to plasma proteins? Is the extent of protein binding either concentration or time dependent? These are important questions because only the free or unbound drug is pharmacologically active. Sometimes the measurement of unbound drug is recommended when interpreting data. In addition, is information on the distribution to cerebrospinal fluid relevant for this drug? Important information that we typically communicate in the drug labeling under section 12.3 include the percent protein bound of the drug, as well as the volume of distribution. Now here I've combined the M and the E. They stand for metabolism and excretion, and they fall under the general category of elimination. You may have heard that term before. Elimination is a broad category that includes metabolism and excretion. So metabolism in technical terms is the process by which lipophilic molecules are converted into more hydrophilic ones, so they can be excreted by the body. And excretion is the irreversible loss of drug from the body. So once it's excreted, it is not coming back. Factors to consider under this section is: how is a drug metabolized? How is it cleared from the body? This usually involves either hepatic metabolism or a combination of hepatic metabolism and renal excretion. And what fraction of the drug is being metabolized? And are there active or toxic metabolites that are being produced? Important information that we typically communicate in the labeling under section 12.3 include the half-life of a drug, the major route of elimination, and the percent excreted in either urine or feces or both. And here I have a few other important considerations for a drug. Is it a narrow therapeutic index product or drug? If it is, that means that very small or slight changes in drug exposure could significantly impact the drug's safety or efficacy, which then may require therapeutic drug monitoring in clinical trials and in clinical practice in order to minimize these toxicities and also to minimize the loss of efficacy. Also, is this drug a significant inhibitor or inducer of either cytochrome P450 enzymes or transporters? If so, this drug may need to be evaluated in further drug interaction studies. And now I'll move on to the next section of my talk, which is giving a brief description of early clinical studies that involve clinical pharmacology. First, I'd like to say a word or two about the timing of clinical pharmacology studies. We're often asked this question about when should I conduct them? Well, the answer is as early as possible. Right after data is collected from preclinical development and an initial IND is submitted that contains the first-in-human study in the US, we begin the start of clinical development. And during this process in phase 1, phase 2, and phase 3, the earlier you can get information from clinical pharmacology studies, the better, because in that way you can help inform the next study or the next phase that comes through, and then that helps maximize the chances of success for your drug. And early phase studies are usually designed to investigate the safety and tolerability, and sometimes to identify the maximally tolerated dose and the PK of the drug in humans. So with all of this information, this is critical for each step in the drug development process. So I mentioned that the start of an IND is the first-in-human study. So usually a first-in-human study entails a starting dose that's determined by some sort of preclinical tox study. And it includes a randomized placebo-controlled study in healthy volunteers. Sometimes patients are used in these studies, but that's fairly rare. And what we're looking to gather from a study like this is some initial safety and tolerability data, possibly the maximally tolerated dose, general PK characteristics of the drug, maybe it's highly variable, what is the PK linearity, and possibly some steady state parameters from the multiple ascending dose study. And preliminary exploration of drug elimination is also ideal. Next, we have the ADME study, which is recommended for nearly all drugs in development. Its objective is to understand the clearance mechanisms of the drug and any and all metabolites in humans. We just put out a new guidance on mass balance studies, and you can find that in the link here, or you can navigate to the FDA guidance website. It's sometimes referred to as mass balance studies, but a lot of folks also call it an ADME study. What it involves is you getting a radio-labeled drug molecule, administering it usually as a single dose to healthy volunteers, and using the intended route of administration, and then in the end, measuring concentrations of the parent and any and all metabolites to determine the relative amount of radioactivity in plasma and other biological matrices such as urine and feces. What we're hoping to learn from this study is to determine the overall pathways of metabolism and excretion for the investigational drug, finding out if there are any metabolites in circulation, and then determine if what the abundance of those metabolites are relative to either the parent drug or total drug-related exposure. Next up, we have bioavailability studies. It's important to conduct these in order to evaluate the rate, or Cmax and Tmax, and extent, or AUC, of absorption of a drug from a test formulation and then becomes available at the site of action. It's typically a crossover single-dose study in healthy subjects where the extent and rate of absorption of the parent drug and any major metabolites, if any, are measured. We can assess either relative bioavailability, which is testing one formulation versus another, or absolute bioavailability, which is the investigational formulation versus the IV formulation of itself. What we hope to learn is the amount of drug that reaches systemic circulation from a tested formulation. And we do have a guidance for industry that was just put out or updated last year on the FDA website. Moving on to my favorite kind of study, a food effect study. Since we all eat food every day, it is important to evaluate the impact of food on a drug's systemic exposure. So the overall objective is to evaluate the effect of food on the rate and extent of drug absorption from a given formulation, and this involves all oral formulations, of course. It involves a single-dose study in healthy volunteers using the highest therapeutic dose of a drug product. In the fed state should be an FDA high-fat, high-calorie meal, although other meals can also be studied. But the FDA recommended high-fat, high-calorie meal is contained in the new food effect guidance, which was just updated last year, and you can find that on the FDA guidance website as well. The PK assessments that come out of a food effects study are very similar to a bioavailability study. No food effect could be concluded if the 90% confidence interval of the fed to fasted ratios for Cmax and AUC fall within the 80 to 125% bounds. If an effect of food is found in a study like this, the clinical significance of it would be determined based on a drug's exposure response profile. What we would hope to learn from a food effects study is how to administer a drug in clinical trials, as well as how to label instructions on how to take the drug with respect to food, eventually in the drug labeling for patients and healthcare providers. So now I'll touch on hepatic impairment studies and just some brief points about it, such as when one should be performed. If a drug is intended to be chronically administered and is systemically available, if hepatic metabolism or excretion accounts for a substantial portion, which is greater than 20% of the absorbed drug, of the elimination of a parent or active metabolite, if it's a narrow therapeutic index drug, and this is irrespective of the proportion that's metabolized, or the metabolism route is largely unknown. If any of these criteria fit the investigational drug, then the FDA recommends that a hepatic impairment study be conducted. Additional details are listed in the hepatic impairment guidance, which is on the FDA guidance website. Speaking of organ impairment, next we have renal impairment studies. When should one of these be performed? Well, if the drug is likely to be used in renally impaired patients, and when we think that impaired renal function is likely to alter the PK of the drug or its active metabolites because they are substantially eliminated by the renal route. If it's a therapeutic protein or peptide with a molecular weight less than 69 kilodalton, or drugs that are likely to be used in patients with end-stage renal disease undergoing dialysis, they should be evaluated both while the patient is on dialysis and off dialysis in order to determine if the contribution of the dialytic method to the elimination of the drug and its potentially active metabolites. These details, as well as others, are set forth in the new updated renal impairment guidance that was just put out 2 years ago, or 3 years ago on the FDA guidance website. So one very important aspect of clinical drug development is the assessment for the potential of a drug interaction with the investigational drug. Now, the investigational drug can be either the drug being impacted, or it can be the drug that's altering the PK of other drugs. So first, we start with in vitro tests or screening to determine if the investigational drug is a substrate for or an inhibitor or inducer of common drug metabolizing enzymes and transporters. These are set forth in the guidances for industry on the FDA guidance website, and we've divided it into 2 guidances, one for in vitro and one for in vivo, and these were just updated in 2020. And some of the more common enzymes or drug metabolizing enzymes are CYP3A4, 2C9, PGP is a very common transporter that we recommend screening for as well. If any of these initial screening tests are positive, we recommend conducting an in vivo drug interaction study to confirm the involvement of the drug, whether the drug is a modifier or it's the substrate. Then, based on the results of these drug interaction studies, it can have some wide implications for drug labeling. These implications can range from something informative such as drug X is not a substrate for CYP3A-mediated metabolism, all the way to a contraindication for the use of this drug with other drugs. In order for drug interaction studies to be informative, they need to be designed very carefully. There are some key points that need to be considered when conducting a drug interaction study. Some of these would be factors that need to be taken into account in order to maximize the possibility of detecting an interaction. So keep in mind that we're trying to put forward a worst case scenario, if you will, so that we can find out if there is a true potential for a drug interaction. So we need to select a proper dose for the inhibitor or the inducer. We need to think about the route of administration, the timing of the co-administration of the 2 drugs, as well as the number of doses to be given of each of the 2 drugs. The degree of the effect, either inhibition or induction of a drug metabolizing enzyme, is typically classified by the change in the substrate's AUC. So it could be a weak inhibitor, a moderate, or strong inhibitor. Here, the exposure response profile again is important for the drug in order to assess the clinical relevance of any change in AUC caused by either an inhibitor or an inducer of the enzyme of interest. So next, I'll just touch very briefly on physiologically-based pharmacokinetics, or you may have heard PBPK. PBPK modeling is grounded in what we know to be true of human physiology, and there are a lot of inputs that go into these models. In theory, This approach allows us to make predictions based on our knowledge from previous trials and previous in vitro investigations, and also sometimes preclinical studies. So what exactly is it? PBPK is a mechanistic modeling approach which utilizes all of these inputs, preclinical, in vitro, in vivo data, to help us predict the behavior of drugs in humans. And what we typically apply this to is to explore the effects of various intrinsic and extrinsic factors on human PK. Some of these factors can include, age or ethnicity, the underlying disease status, or even sometimes drug interactions. So another method we use in order to make some predictions of drugs without directly and empirically testing it is model-informed drug development. And I guess in technical terms, PBPK can actually fit under this larger umbrella of MIDD, because MIDD is an approach that involves developing and applying exposure-based biological and statistical models derived from preclinical and clinical data sources to inform drug development or regulatory decision making. So it's a pretty broad umbrella. And also having well-timed and well-designed dose-ranging studies or dose-finding studies are important for avoiding dose selection pitfalls later in development, and MIDD can help with these dose selection type of issues. A few years ago, the FDA initiated an MIDD paired meeting program that allows drug sponsors and drug developers to meet with the review team, which is, in the case of MIDD, led by clinical pharmacology. Some of the focal points or issues that have been discussed in the past have been dose selection or estimation, clinical trial simulation that's based on drug trial disease models. These may be used to inform the duration of a trial or even selecting appropriate response measures, predicting outcomes, etc. We've also had discussions about predictive or mechanistic safety evaluations. In these cases, we've used systems pharmacology and other mechanistic models for predicting safety, and also identifying critical biomarkers of interest. So we've had a lot of successful cases of MIDD already. And last but not least, I'd like to acknowledge Kelly Reynolds for the use of some of her slides, and Leonard Sachs for the invitation to do this presentation, as always. Thank you.